Okay, fine, fine.
Okay, sir, मैं पहले कहता था हम अपने इंट्रोड्यूस करते हैं.
Okay.
Yes, yes, yes. Okay, thank you. So we'll start the next session. The next session will be conducted by Dr. Satyasai Jagannath Nanda, who is an associate professor in the Department of Electronics and Communication Engineering at MNIT Jaipur. He received the PhD degree from School of Electrical Science, IIT Bhubaneswar in the year 2013, and MTech degree from Department of Electronics and Communication Engineering from NIT Rourkela in the year 2009. Sorry. He was the recipient of Canadian Research Fellowship from the Department of Foreign Affairs and Intern Trade, Government of Canada for the year 2009 and 10. He was awarded the Best PhD Thesis Award at SOC PROS 2015 by IIT Roorkee. He received the prestigious IEI Young Engineer Award by the Institution of Engineers, Government of India, in the field of electronics and telecommunication engineering for the year 2018 and 19. Dr. Nanda participated as a young scientist in the second SCO Nations Young Scientist Conclave 2023 at Jawaharlal Nehru Center for Advanced Scientific Research, Bengaluru. He is a senior member of IEEE and IEEE Computational Intelligence Society, a long-term member of Soft Computing Research Society, New Delhi. Till date, he has published 60 ACI journal articles, 65 national conference proceedings, one book chapter, and he has edited 6 book proceedings of Springer, which till date has received more than 2,200 citations, and he has also received the Best Paper Awards at IEEE Odicon 2022, which was happened at SOA University, Bhubaneswar, and also received Best Paper Awards at various conferences such as SMIT, SOC PROS, OPCON, and OWT 2017. He is the in charge of Digital Signal and Image Processing Lab at MNIT Jaipur. He is the lead guest editor of Scopus Index Journal, SN Computer Science Springer. He is the principal investigator of the 5G laboratory to be established by Department of Telecom, Government of India at MNIT Jaipur. Under his supervision at MNIT Jaipur, 8 researchers have been awarded PhD. 5 researchers are continuing their research. Along with it, he has supervised 32 MTech theses and 15 BTech projects. Dr. Nanda is the coordinator of Electronics and ICT Academy at MNIT Jaipur. And this program is also funded by E&ICT Academy. So Dr. Nanda's research interests include adaptive signal processing, seismic signal processing, multi-objective optimization, and image processing. So with this, I request Professor Nanda to please start the session, sir. Thank you so much.
Thank you, Dr. Deepak, for giving an introduction. So let me share the slides. Uh, my screen and audio all are fine. So shall I go ahead?
Yes, sir. You can start.
Okay. So today I am going to discuss about the application of this one got disconnected, I think. Yeah. This one got disconnected. Just a minute. Yeah, this one got power. fine. I believe no problem over here. I can. Yeah, the audio. Okay. Is the audio is fine? Audio and video. Okay. So do let me know if there will be any internet connectivity or anything in between. Then do let me know. Uh, so today we are going to discuss about unsupervised learning algorithms for medical image segmentation. In this particular, we are going to focus about determining a segment of an MRI image, which is basically focusing on various kind of tumors developed in the brain. And one of the typical disease is glioma tumor. Okay. It is a kind of tumor that is mainly localized in the brain and caused by the glial cells. This tumor is generated due to three types of normal glial cells: astrocytes, oligodendrocytes, and plethemical cells. So with these three type of glial cells, these tumors get generated, and you can find these particular type of tumors data at the Harvard University website. I have highlighted over here. So the glioma tumor brain image can be obtained considering the different type of categories. And the first one is the glioma FDGPT image technique. And this is the MRI image you can able to observe. The 2D image that has been generated out of the position emission tomography. Similarly, another type is given over here, which is generated from single photon emission control tomography technique. And this today 2D image from different angles is different in SPECT imaging technique. And the third type, you can observe that this is a type of tumor that spreads from one part of the brain's position to another particular position. Uh, and these cancer cells travel through the bloodstream, and that's how it can infect from one particular organ to the other organ of the body. The fourth particular type is a type in which the malignant uh, grows inside the brain, and it can inflate other tissues which are nearby. The fifth one over here, you can able to observe that this is known as sarcoma brain image, MRI image, and they are basically categorized into two type of groups. One is bone sarcoma and other one is soft tissue sarcomas. So these all 5 type of various brain tumors I have shown you. Our objective is to determine the location of the tumors in this particular images. Accurately determining them is really a challenging problem. Over here, we are going to apply unsupervised learning algorithms, and one of the most prominent learning unsupervised learning algorithm is known as clustering. In clustering, we try to do uh, identify similar kind of patterns or similar kind of data to be or homogeneous data to be together in a particular group or in a particular cluster that is different from other type of segments. Over here, you can able to observe that a typical simple example I have taken of 3 different flowers like Setosa, versicolor, and versinica. We have 150 observation, and each one of them is represented over here in blue color dots, as you can able to observe. So in this particular thing, without clustering, this is the data set. And when we apply the clustering, you can observe that it has been partitioned into 3 different categories with pink color dots. They are completely isolated, whereas the red and black color dots, they are overlapping in nature. So this overlapping and non-overlapping is a challenging task in the cluster analysis, why there is a overlapping between black and red color dots, because this versicolor and versinica, these 2 flowers look alike, whereas the setosa flower looks completely different. So it is very difficult when you have 2 different type of groups and they are alike and we want to distinguish them into 2 different segments. So under that circumstances, the overlapping portions comes into account. Our objective is to cluster or group these particular data in a efficient manner so that the overlapping can be avoided as much as possible. So the image segmentation is formulated over here as a clustering problem. And in clustering, we can able to deal or formulate this particular one as an optimization task, in which our objective is to optimize similar kind of pixel in various groups. Okay, or similar kind of pixel values, we want to group them together so that we can determine the nature or characteristics of the tumor growing inside the brain. So for that one, what we have to do, every pixel value, we have to, in the entire image, if we want to partition it into k different segments, then we have to initially take k number of centroids. And from each centroid, we have to determine the pixel values differences from the center, and then try to minimize that which pixels are closer values to those central values. So accordingly, the algorithm will proceed to divide the entire uh, image into various portions, in which some portions may be containing the tumor and in some portion the tumor will be absent. And uh, various kind of statistical techniques we can able to use to uh, break down the entire image into groups. And one of the technique is minimization of Euclidean distance, in which we perform k-means based cluster analysis. So this in this kind of problem, you can observe that over here a data set or an image set, you can say, which has n cross d dimensionality, If we are considering a 2D image, then it will be n cross 2. Okay. And we are trying to divide this particular image into multiple groups. Okay. And each group represents one particular characteristics of the image. And we have to partition the groups in such a way that every group must contains at least some pixel values. There should not be any cluster or any particular segment in which there will be no pixel values will be there. Such type of partitions is not admissible. There should not be any common pattern between any two particular segment. That means one particular portion, we cannot able to say that it is both, it contains both tumors as well as normal cell. So that will not be there. So interconnection between the two portions should be phi. That means tumors will be separated and non-tumors or regular cells will be in another cluster. And if the image set comprise of multiple number of segments, then summation of all the segments should be equal to the entire cluster. So considering the partitional clustering, there are other type of image segmentation techniques have been reported by several researchers over the year. And the other popular techniques are hierarchical clustering based techniques and fuzzy based clustering techniques. In hierarchical clustering techniques, you observe that between all these similar kind of pixel values, a tree-like structure has been grown from bottom to top, and that is known as a dendrogram plot. And over here, when you cut this particular dendrogram uh, in the form of a line, you can observe that four different cluster or segments have been reported over here, like the green, the red, the blue, and the sky color one. And the popular algorithm based on the hierarchical clustering are allomorative algorithms and divisive algorithms. There are further the fuzzy based clustering techniques are there in which every particular pixel values is uh, connected to all the segments with a probabilistic manner, and the pixel values will be assigned a probabilistic value. So based upon this particular thing, if we partition a data set, if you will observe over here, the green particular portions have probabilistic values much higher for the greenery things, whereas it is low for the blue and the violet color. And the fuzzy partition can be converted to crisp partition by considering the highest value of the statistics into account, or highest value of the probability into account. So considering this, the popular algorithms are fuzzy C-means clustering algorithm FCM and fuzzy C-cells algorithm. The other one, partitional clustering, which I discussed with you, the popular algorithms are k-means, k-medoid based algorithms, in which from the centers the Euclidean distance is considered and various portions have been segmented. So in k-means based clustering algorithm, k stands for how many partitions we want to make, and mean stands for the central portion of the cluster location. Cluster center should lie considering the mean of the pixel values or the average value of the pixel values for obtation of the centers. So over here, if you observe that if we have some pixel values like this given here on the left topmost side for white color dots, here we have divided the data by considering two important points. One is this red color triangle and another one is yellow color triangle. From every particular point, we have tried to determine the distance, the minimum distance we have tried to find it out, that which points are close in distance to this red triangle and which points are close in distance to this yellow triangle. So based upon that, we have tried to bifurcate this entire data into two different partitions. And once the initial partitions have happened, then we have further moved these particular location of the triangles into the central locations by considering the means. So based upon this, after a couple of iteration, you observe that the triangles are lying on the central plot of these two groups, and these groups have already been bifurcated effectively, and they both the groups represents two different segments of an image. So this is the steps associated with k-means algorithm, and we can take Euclidean distance as well as Manhattan distance based upon which type of image portions we are dealing with. And this is the mean calculation based upon which the updation takes place. So the k-means clustering algorithms are effective, and they are there for longer duration of time. And in these days for big data analysis and video analysis also, the k-means algorithm plays a very vital role because of their simplicity or lower computational aspects. But these algorithms always formulate spherical shape cluster because of the calculation of Euclidean distance in the form of circles. In most of the applications, these clusters may not be spherical in shape, but in some times they may be arbitrary shape in nature. So for that particular type of application, instead of going for k-means based algorithm, the density based partitional clustering algorithms are more suitable for such kind of applications. So k-means is basically a hill climbing based approach for the optimization. It takes care into account, but in many other cases, the optimization can be formulated uh, or the fitness function may not be a unimodal function like that can easily be achieved a maxima or minima considering one particular optima. In many cases, the statistics or the function we are optimizing uh, for the cluster analysis is not a unimodal function but is a multimodal function in which there are multiple maximas and multiple minima. So in these cases, the traditional derivative based approaches or hill climbing based approaches do not give us effective results. So the global minima or global maxima we cannot able to determine by considering such circumstances. So the output obtained will be a local maxima, or effective partitions cannot be obtained for such kind of problems. So for this, the derivative based traditional approaches like the steepest descent, conjugate gradient, Newton-Raphson based methods, and all these methods do not give us effective results. So we have to build clustering algorithms by considering the nature-inspired optimization algorithms or evolutionary based optimization algorithms. So in such kind of algorithms, the birds, ants, molecules, natural species, they combine together to formulate a group which can solve the optimization problem, and the clustering is dealt over here as an optimization task. So if some of the elements you can observe, they get stuck to the local optima, the rest of the elements, there is high probability that they can go to the global maxima. And once they reach the global maxima, they will inform the other particles so that they can also change their position and all the particles will move to the global maxima. So such type of statistical problems, I told you that they have multiple modes are egg holder, drop wave, alpine, Greenwich, Rastigen, De Jong. Such type of benchmark functions are there for simulation. And the real life uh, application to the clustering can be observed over here that the evolutionary or nature inspired based algorithms have been reported for these cluster analysis problems from for last 2 decades. And the various algorithms reported are genetic algorithm, evolutionary strategies, genetic programming, differential evolution. You can observe that I have given over here a table which is comprising of both single objective and multi-objective clustering techniques which have been applied by various researchers. In evolutionary algorithms, what happens that from the parents to the next generation children's, uh, the solutions become normally better, and the betterness continues to improve from generation to generation. Whereas in swarm intelligence, it is not just the parents, it is a group of people's collective intelligence to swa- behave properly to determine the food location. Like a group of birds, they are searching for food and they all move in a particular manner. And a group of ants, they all move on a single line to collect their food from the source to the hole. Based upon that, many algorithms have been reported by researchers over the years. And there are also other biological phenomena from which these algorithms have been inspired, like the artificial immune system based algorithm, bacterial foraging based algorithm. They do have their own principle for update of positions, and the corresponding algorithms, single objective as well as multi objective versions, have been reported by the various researchers. The main thing about the single objective difference between single objective and multi objective is we here in single objective optimization we are considering only one particular criteria, like the minimization of Euclidean distance or the maximization of a Silhouette index or similar kind of thing. We are considering only one function for the cluster analysis task. Whereas in multi-objective optimization, we simultaneously deal with two or more statistical functions which are contradictory to each other, and uh, they give us an effective balance of the clustering. So among the most popular swarm intelligence algorithms, the fundamental algorithm is particle swarm optimization, which was reported by Kennedy and Eberhard in 1995. And here a swarming phenomenon is shown in which if we are moving to a particular pond and throwing some food particle, the fish which is close to this particular food particle can observe this food, and that will estimate how much amount of food the fish has got. It will move there and give a signal to its friends. And all of its friends who are in nearby locality, they will move towards the food. So this particular phenomenon is mathematically formulated in the form of an algorithm like this, that this circle is representing a pond in which this particular violet color circle representing the food, the xi1, xi2 up to xi5, these all represents the particle positions and the corresponding velocities. And after some times, if you will observe that all particles are changing their positions and they all are moving their food, moving towards the food after certain time. So this is in the third thing, you can observe that all particles are moving towards the solution. So this is mathematically represented like this, and the updates are given over here like the local best position represents every individual particle, how they are acquiring their past experience and the present experience. The global best position deals about the best location of a single particle which is best among the swarm or a group of fishes from there which particle or which fish has obtained the particular best solution, that is the global best position. So the velocity and position update equations have been narrated over here, where the updated velocity is comprising of a weighted version of the previous velocity and a difference of the local minus the present position and the global minus the present position. So every particle gets attracted towards the local best solution and the global best solution for its velocity update. Once the velocity get updated, the position automatically get updated, you can observe from the second equation. So when dealing with PSO for different applications, the velocity and positions has to be restricted. If we do not restrict, then the solutions will move out of bound, which will not give us an effective result. So this is the PSO update equations. And like the particle swarm optimization, there are many more algorithms like the genetic algorithm, differential evolution, ant colony optimization, ABC algorithm, artificial immune system, bacterial foraging optimization. All these algorithms have their own steps. I have summarized their steps over here, that how you can able to move from with each particular algorithm steps, iterative steps to determine effective clustering solutions for your problem. Like I told you that the minimization of Euclidean distance is one particular criterion. Similarly, there are other statistical measures which you can employ as a maximization or minimization task to do the cluster analysis. Here you can observe that midway distance minimization, centroid distance minimization, intra and intracluster distance, either their maximization or minimization, variance ratio criterion maximization, Dunes index maximization, CLOT index maximization, CS measure minimization, all these things, statistical measures, either maximization or minimization function will give us effective cluster analysis values. And these are the researchers, they have used these particular fitness functions in last years for effective clustering. This is a survey article which I wrote around 10 years back in 2014. And till date, this single article has received more than 650 citations. So those who are interested about the image segmentation and the cluster analysis algorithms and their application, they can refer this particular review article. Now I want to discuss with you about another swarm-inspired algorithm based on social spider algorithm. This is a concept which is little bit different than the particle swarm optimization, because in particle swarm optimization, the weight update equations are directly related to the vector summation of the difference between the global best position and the local best position. The vector sums are linear added summations, but here in this particular algorithm, the update equations are nonlinear in nature. So that's why we are moving towards this algorithm. So in this algorithm, it is also a multi-agent algorithm compared to the particle swarm optimization. Multi-agent means here I want to define that this is a swarm of spiders in which there are male spiders, female spiders, and baby spiders. They all have their own weight update equations and they all work together to give better results for us. So initially, while doing the initialization in particle swarm optimization, we have to initialize all the particles. Whereas over here, we initialize 60% as female uh, spider and 30%, 30 to 40% as male spiders. So two different search agents we initialize, and the update equations takes them accordingly. You can observe that the fitness evaluation is taken as minimization of Euclidean distance between the data points to the cluster center, and the cluster centers represent the spider positions for the cluster nodes. So accordingly, we can assign the weights to the spider, which is a function of the fitness value, and the population is further fragmented into two different categories, that which populations is more than the median weight, and otherwise it is lower than the median weight. So the population is segregated into further two types. So this particular equation I wanted to mention that the one the velocity of the local and the velocity of the global, they all are function of e to the power minus delta SISL square. So this exponentially decaying function that introduces nonlinearity. It is not the straightforward movement, but it is a exponentially decaying thing, which is nonlinear in nature, and the weight updates get affected with this particular nonlinearity. And with this, the spider movements becomes much better for the weight update uh, uh, position. So this is the female spider movement. You can able to observe here the update equations. The update equations are basically of two type. One is if the random number is lower than a probability factor, the upper equation gets satisfied, and otherwise the lower equations get satisfied. The difference between that, in upper equation there is all things are positive, while in the lower equation the all coefficients are negative. So a spider can either move forward or backward, depends upon this particular update equation. Here the spider is moving forward, and it is used for the exploration of the space in the entire wave. And then moving on to the next process is for the dominant males update equation. You can observe that this update equation is different than that of the female spider update equation. And the third weight update equation over here, you can observe that this is for the non-dominant male spiders update, in which this is helpful for the exploitation of the search space. That means deep navigation on a particular location of the search space for the weight update. So over here, further the mating operation takes place, in which the probability calculation is done and the new baby position is taking the highest percentage factor on which the mother's location is there. Here it is 36% you can observe for F2, on which the new spider's location gets generated. So after applying this particular thing, the original algorithm was containing this female position update, dominant male position update, and non-dominant male position update in sequential manner, one after the other. And in our proposed algorithm, these all operations have been carried out in parallel manner. And we have applied this one first on the benchmark datasets, like the iris flower dataset I discussed. Along with the iris flower, there are other small dimensional and high dimensional dataset, like the KKD COP dataset we have applied, in which more than 1 lakh samples are there with 10 different classes. So you can observe that the classification error percentage are lower compared to the k-means real coded genetic algorithm and PSO based algorithms. This particular algorithm is giving us better effective results. And this is the results on the KKD COP bio dataset with 10 different clusters. And here you can observe that the topmost corner result is giving us better result because there is lower overlapping between the samples of the dataset. So we can observe that in these datasets, the computational time is lower due to the parallel operation incurred. Like 25.06 second we have observed for Statlog dataset compared to 44-second time with the original SSO algorithm. Similarly, for the dermatology dataset, 1.41 second instead of 1.462 second. So every time the computational time is lower, and over here we can observe the results of the glaucoma brain MRI image dataset, in which the segmented images uh, are given over here, that the segmented image with the proposed algorithm on the top right, and segmented images with the genetic clustering and the k-means clustering algorithm as also included over here for all the 5 data sets. You can observe that the second one, third, the fourth, and the fifth dataset, in which in all the cases the segmentation results are effective compared to the k-means and the genetic clustering algorithms. Here the clusters are very effectively identified, which is representing the tumor and non-tumor partitions. So the various other statistical techniques like normalized root mean square error, PSNR, SSIM, and Tohu's coefficients measure have been reported over here for all the 5 datasets, in which the algorithm shows superior performance. So, like one particular algorithm, here it is a many objective optimization algorithm, NSGA3, is reported by Professor Kalyan Mahdev of Michigan State University, in which we can effectively use multi and many objective clustering techniques for analyzing such kind of data sets. So those who are interested, they can go through this particular algorithm of multi and many objective optimization uh, of and they can able to download the code from the Michigan State University website as well as from the IIT Kanpur uh, uh, website link of Professor Dev, and they can apply this particular algorithm for also effective image segmentation. So 5 different type of glaucoma and brain tumor images are segmented by the proposed algorithm, and they have shown better results compared to the genetic algorithm based clustering and k-means based clustering algorithm. So some of our related publications you can see in these particular websites. And thank you for this much from my side for presentation today. Yeah, so if participants have any queries, we can able to see in the chat box. Oh, stop sharing. Just can I take it, please?
Can we access the recording sections? Okay,
Okay, what type of clusterings are density-based clustering? Like DB-scan means density-based clustering always takes care about uh, a specific portion of the segments in which there are dense areas and there are other portions in which there are sparse areas. So dense areas will be clustered differently and sparse areas will be clustered in other different manner. So different groups can be formulated accordingly, and many density-based clustering algorithm have been reported by researchers in last several years. And you can go through that particular density-based clustering algorithms. There is another research paper from our group. You can observe that we have also proposed some low computational complexity-based density-based clustering algorithm, and a lot of development have been reported on that particular cluster analysis-based algorithms. Uh, Obviously, those things we have applied for the seismic clustering analysis, and similar kind of algorithms can be applied for biomedical image analysis as well. What kind of clustering method is suitable for high dimensional cases? Because I learned somewhere that Euclidean distance is not suitable matrix for high dimensional data. It is not always about the which particular distance matrix you are taking. Actually, the computational complexity of overall algorithm always matters. So if an algorithm is very complex, then you are applying on high dimensional data set, it will take a lot of time. And if it is an offline application, we can able to afford the time and get the result. There is no problem. But if it is an online application, then there will be a problem because high computational algorithms cannot be effectively applied on high dimensional data set on which quick results are required. So that is the challenge. PPT we will share over the time, and can I see some storage chart? These two are the questions. Yeah. Is there any, is there a difference in relation in applying segmentation for clustering and for? Is there a difference in relation to applying segmentation for clustering and for classification? Segmentation, image segmentation is formulated as a clustering problem over here. What I have explained is image segmentation is formulated as a cluster analysis problem. Classification is different. So image classification is different. Here, a single image, I am partitioning into various segments. That is the thing. One particular tumor image and brain image with tumor, that one we are partitioning into various segments and trying to identify the tumor locations. The data set collected from any organization for this study. So these brain images I have taken from Harvard data set. So Harvard University dataset I have given in the front slide on the beginning slide. So my slide, Dr. Deepak will share with the participants, and on that one already the dataset things is given. Any other thing? Any other queries? Sure. Okay, okay. For classification,
segmentation be used to determine no.
look, for classification purpose, can segmentation be used to determine different classes? No, no, no. So segmentation is a different problem in which one image is segmented into various portions, and each portion has its own importance. Classification purpose is means one particular image you are giving, that particular image is belongs to some type of disease or not belongs to some type of disease. That is a classification problem. Try to understand the fundamental difference between these two. Suppose somebody is cancerous or non-cancerous. So this is a classification problem. Many images we give, and these are cancerous image, these are non-cancerous image. But here we are not doing that one. In segmentation, what we are doing, we are trying to give one particular image. That particular image will be bifurcated into various segments and identify that which location is tumor, which location is some other portion. So like that, we are trying to find out the significance of various portion of a single image. That is what image segmentation. Various segments we are trying to analyze of a single image. Hope I have answered your queries. Any other query? In some literature, segmentation is used before feature extraction. If we use this optimization technique to find region of interest, no, will it be computationally intensive? Yeah, it may be computationally intensive compared to PSO. Because PS particle swarm optimization is very simplified algorithm. That's why everybody prefers PSO. But this algorithm is giving comparatively better result compared to PSO, but with some increment in computational complexity. That we agree. Is this essential to combine feature extraction before segmentation stage? Feature extraction, not necessarily. If there is some noise, you can remove noise and then do feature, then do image segmentation. But no need for feature extraction before image segmentation. Can explainable AI implemented on segmentation? Yeah, means the model can be used for image segmentation, but for that one, there will be separate session for explainable AI in this particular FDP, in which the expert speaker of explainable AI will discuss about this particular problem.
Okay, so thank you very much, sir, for the wonderful talk. So it was truly inspiring how to use these optimization techniques for solving medical image clustering problems. So basically, medical image segmentation problem using clustering techniques, which is unsupervised one. We do not need even ground truth in this case.
No ground truth from direct image we can able to do it.
So that is the most important, I can say, the point of using unsupervised
Yeah.
based learning
When
techniques.
you do not have any target data, you can able to do this,
You will-
analyze and get the results.
Absolutely. So thank you so much. I hope participants have already got the idea behind the usage of clustering techniques to solve image segmentation problems. So thank you once again, sir, for
Thank-
the wonderful talk, sir.
Thank you, Dr. Deepak.
Thank you.
Thank you.
Okay. So participants will start the next session at 7 p.m., right? So let's have a 12 minutes of break and then we'll start the hands-on session, right? The first hands-on session at 7 p.m., sir. Thank you.